And this is where we left off, just explaining what an oxidation reduction reaction is. With regard to methane at the bottom, what you're looking at is methane loses electrons, becomes carbon dioxide, so it loses electrons, it becomes oxidized. Oxygen gains electrons, becomes water, it's reduced, energy is released. And this is methane is a cooking agent. Methane's a gas that we use many times to cook our food. So it's another reaction that can be considered oxidation reduction reaction. Now you might be asking yourself, why do we go to all this trouble? And that is because that electron then has so much potential energy. There's so much energy stored in that electron. Actually, it's way too much energy stored in that electron. So what the body has done in terms of evolution is there's this cofactor, coenzyme, NAD, that which is NAD is nicotinamide adenine dioxide. I'm actually I'm saying that incorrectly. We always talk about it as NAD. NAD is nicotinamide. But the important thing about it is it has the capacity to carry this electron. So it is useful in part of glycolysis. It's created in glycolysis, but it's created because it's useful to carry this electron. So in cellular respiration, glucose and other molecules, organic molecules, are broken down as a series of steps. The electrons from organic compounds are usually transferred to an electron carrier, and NAD happens to be one of them. It's a coenzyme. It's a derivative of what we consider vitamin B, which is niacin, and it easily recycles between its oxidized and reduced form. That's why it's called an electron carrier, because it will carry that electron, but it'll also give it up. What it does, what these coenzymes do in the electron transport chain is pass the electron on down the line. Each time it gets passed down the line, it loses a little bit of energy. And so the energy loss is at a very controlled rate. In terms of NAD, then becoming reduced in its reduced form and having that hydrogen ion, atom bonded to it, you have that stored energy is trapped onto that electron carrier. Remember, with regard to covalently bonded organisms or covalently bonded compounds, it's still going to be oxidation reduction, but we think of it in a form of a hydrogen atom. And so NAD is the thing we're introducing here. It is the electron carrier. It will show up again when we talk about the electron transport chain. And the first step of cellular respiration makes a little bit of ATP, but also makes this cofactor, this electron carrier. It passes electrons, so NADH, remember that's the reduced form, has a hydrogen atom on it, passes the electron to the electron transport chain. Now understand that it is the whole atom, but it's the electron that we're referring to. And this is the graphic about this electron transport chain is a series of steps. So it's a series of steps down a energy gradient. So you're going from a high energy to a lower energy in little manageable ways. And so it's a series of steps instead of one explosive reaction, and that's what you're seeing in the graphic. On the left, the uncontrolled reaction. If in fact all of these electrons were released at once, you'd have an explosion. And that wouldn't work so well in the body, but because of NAD, this electron carrier, and its capacity to be oxidized and reduced over and over again, you actually have this electron being passed down from NAD to NAD to a lower and lower energy form with energy released. That released energy then in another step, all of this is happening in the mitochondria, can be used to create ATP after a fashion. The O2 pulls the electron down the chain, and one of the reasons that we include the O2 pulling it down is because eventually this electron is going to get together with oxygen and form water. Remember, water is one of our byproducts. We breathe out water as well as CO2. The transport chain consists of another proteins built into the inner membrane. So the NAD eventually is going to pass that electron off to a protein that is in the inner membrane of that powerful mitochondria, that organelle that was taken into a cell many years ago in a symbiotic relationship, and these proteins embedded in the inner membrane is what hold these electrons in a controlled form and allow the energy to be released or to released in stages or fall down steps. The stages of cellular respiration, as I said, there's 3 of them, and we've mentioned them already, but here's a repeat of it. You have glycolysis. Glycolysis happens in the cytosol. So you're seeing glycolysis in the blue block. Glucose is broken down into pyruvate. Glucose is a 6-carbon sugar. It's divided in half into pyruvate. So glycolysis is the partial breaking down of glucose into 2 3-carbon molecules. So a 6-carbon glucose into 2 3-carbon molecules. The citric acid cycle, which is also known as the Krebs cycle, completes the breakdown of glucose into 6 molecules of CO2. So we have 6 molecules of carbon dioxide plus the electrons that are being carried via NADH and FAD, which is also another electron carrier, and actually a very, very much more efficient electron carrier, to the electron transport chain. The electron transport chain is also called oxidative phosphorylation. So the electron transport chain is associated with oxidative phosphorylation. So the chain itself is the falling down of electron energy levels. The oxidative phosphorylation is another step that we'll look at that actually causes that 3rd phosphate to be put onto ADP. ADP to have that 3rd phosphate to turn into ATP. This process, so remember, we begin this process in the cytosol with glycolysis. So the portion of the 3 steps, whether it's glycolysis, whether it's the Krebs cycle, or whether it's the electron transport chain, the largest portion of ATP is produced in the last portion, the oxidative phosphorylation. And it's because of these redox reactions where you have a tremendous amount of shifting. Remember, every time there's a shift or a change in bonds, there's going to be a loss of energy, and that energy then can be used to fuel another reaction if need be. Smaller amount of ATP is made in glycolysis. And by the citric acid cycle, by this process called substrate level phosphorylation. And what's happening here in terms of this image is you have an enzyme that then is going to bring a substrate and a phosphate. So that substrate, you can think of that as ADP, or it happens to be substrate PEP. You actually see it pyruvate. The enzyme is pyruvate kinase. The substrate is abbreviated as PEP. It actually has three carbons, 3 oxygens, and 2 hydrogens, and one of those oxygens has a negative charge on it. That then is going to, one of the oxygens has a phosphorus hanging off of it. That is going to bring that particular substrate together with ADP. You can see this, I'm referring to the image on the right, and that because it's an enzyme, enzymes facilitate reactions. It's going to facilitate the phosphorylation. So facilitate ADP becoming ATP. So this occurs in enzyme transfers a phosphate group from this, the substrate, which is PEP, and that's why this is called substrate level phosphorylation. Glycolysis, a little bit more about that happening outside the mitochondria. So one of the things you want to put in your head is that glycolysis happens outside the mitochondria. One glucose molecule will lead 2 pyruvates. It does not require oxygen. It's pretty universally found in most organisms. It likely evolved into the, it evolved before the citric acid cycle and oxidative phosphorylation. So it generally is older. It's a more primitive process, you might think, because it's not quite as efficient. It happens in 2 major phases. You have your energy investment phase and your energy payoff phase, and we're referring to ATP as the energy that's investing and being paid off. So if we look at this, remember this is happening in the absence of oxygen, in the cytosol. Glucose is broken down into 2 pyruvate molecules, and 2 ATP are used in order to do this. And so then what you have is in the course of ATP being produced from those pyruvate molecules, you have the payoff. You actually have 4 formed, plus this electron carrier. So you have 4 formed in the payoff of the breaking down of that glucose molecule, and that means you have a energy payoff of 2 ATP, plus this electron carrier, NAD, which is the biggest payoff with regard to glycolysis, which then can go into the citric acid cycle. The pyruvate also, the breaking down of pyruvate, that also then is is initially going to go into the citric acid cycle. It'll lose a carbon before it does, but the glycolysis, which is not as an efficient a process, is very necessary because it makes the raw products that go into the citric acid cycle. The oxidation then after glycolysis, pyruvate, which is that 3 carbon molecule, is oxidized to another molecule called acetyl-CoA. With O2 present, when O2 is present, oxygen, pyruvate then will enter the mitochondria almost as a carrier. Oxygen pulls it in. You actually have a transport protein. It's not actually oxygen pulling it in, but that oxygen has to be in the mix, has to be present. It's transported from the cytosol into the mitochondria. Pyruvate then has to be converted from pyruvate to acetyl-CoA. What that does is the carboxyl group, in this image, the gray group, is given off as CO2, and then the carbon fragment is oxidized and electrons are passed to NAD. And we need to do this on the next one, sorry.